and I'd like to thank everyone for coming out today. As a computer scientist at Lawrence Livermore, I think a lot about how we can use computers to better understand the world around us. And today, I'll be talking about how we can use computers to better understand the brain. I got my start in computers when my family got its first computer. I think I was in 3rd or 4th grade. I learned how to program it, and I was fascinated by the idea that one machine can do so many different things. And I carry that with me today in my work when I think about how to use computers to better understand many things around us. Today we'll be talking about the brain. We'll be talking about how it works and what it's made up of. We'll also be talking about computers. So on the bottom here, this is a picture of one of the largest and most powerful computers in the world. It's at Lawrence Livermore National Laboratory. It takes up an entire space on the floor of the building, and inside these cabinets are shelves that contain the various components of the machine. But if we want to use the computer to understand the brain, we first have to represent the brain in a way that the computer can understand it. We have to put the brain inside the computer, and I'll show you how we're going to do that in this talk. But first, let's talk about computers. Computers are binary machines. Binary means 2. All of the data that's processed in a computer has to be represented in terms of bits or binary digits. And you can think of a bit as either a 0 or a 1. There's 2 possible values. So for example, when you take a picture of your cat, it's represented as a very long sequence of 0s and 1s. We're showing just a small fraction of those that can fit on the screen here. And all of the complicated operations that you want to do with this image, like apply a filter to it, maybe change the brightness, is broken down into very simple operations on these 0s and 1s. I'll also be talking about a unit called a byte, where a byte consists of a sequence of 8 bits. So here I've broken down this sequence of bits into groups of bytes, groups of 8 bits. Now let's go back to our original question. We want to represent the brain in a way that the computer can understand it. Why would we want to do this? Well, as I hope you'll appreciate in this talk, the brain is immensely complex, and we need computers to help us understand how it works. I'm going to start by talking about the brain. The brain is the control center of the body. Just like you'll see in the movie after my presentation, the brain receives information from all the parts of the body. Just like in this picture, you can imagine information coming into all these screens in this room from various locations and satellites maybe, and the people in this room have to take in all that information and decide what to do, whether to launch another satellite or move the spacecraft. And it's the same kind of thing inside your body. The brain collects information through the peripheral nervous system, which is depicted with the blue lines in this figure, from almost every point on your body. That information is transmitted to the brain through the spinal cord, and then the brain decides what to do on the basis of that information, and then sends signals back. So for example, if you touch your finger into a hot stove, the nerves at the end of your finger will send a signal through the spinal cord to your brain. Your brain will then decide to move your arm. So it'll send a signal back, contracting the muscles in your arm so that you can pull your finger away from the hot stove. Your finger can't move away from the stove until all of that happens. So the signal goes up to the brain and back down. And hopefully it happens fast enough before you get burned. The brain controls many of your organs without even knowing about it. Your brain controls your breathing on a regular basis. You can, intervene and, for example, take a deep breath, but the regular operation of breathing is controlled by the brain. The heart can actually function, beats entirely on its own without any input from the brain, but the brain can help modulate it. It can help increase the heart rate through, for example, adrenaline when you're exercising. But what is the brain made up of? Well, the brain is made up of many different structures. But the one that I'm going to talk about most of all is a cell called the neuron. And the neuron is a cell just like any other cell in your body. Inside the cell body is the nucleus containing the genetic information. Then attached to the cell body are these tree-like structures called dendrites. The dendrites are the input to the cell. Other neurons will attach to this neuron through its dendrites. There's also a long tail-like structure called an axon. And I'll explain later in my talk, but on occasion, a neuron will send a signal or a spike through this axon. It then goes through the axon terminal where it is transmitted to other neurons, to the dendrites of other neurons. Neurons can be small, but they can also be very long. So you can have single neurons that connect different areas of your brain, and they also make up the nerves in your body. So the longest single cell neuron in your body goes from the base of your spinal cord down to your feet. So now I'm going to talk more about the brain in terms of its structure and its function. So in this analogy, we can think about structure and function of the interstate system in approximately Northern California. So on the left, you see the map. The structure of the roadways is the precise location of each road and which other roads it connects to. If I want to go to Sacramento from here, I have to take, I think, 205 west and then 5 north. In order for me to do that, in order for me to know which roads to take, I have to understand the structure of the interstate system. The function, on the other hand, would be the traffic at any given point in time. So here the picture on the right is showing you the terrible traffic conditions in the Bay Area. And the function of the traffic can change quickly over time. Whereas the structure doesn't change. Or if it does change, it takes a long time for it to change. It takes a long time to build a new road. As another example, an electronic circuit. So on the left, you can see an image of this circuit. And the structure of this circuit are the individual wires, or the individual traces that connect the various components of the circuit. Whereas the function, depicted on the right, are the electrical currents that are flowing through those wires. If it's a computer, maybe the zeros and ones, the binary digits that are flowing through that circuit. In the brain, the picture on the bottom left is called an electron micrograph. That's like a very powerful microscope that can let you see individual neurons. So in the brain, the structure is the particular pattern of which neurons are connecting to each other. And the function, is depicted on the right, is the activity of those neurons. Because the neurons are sending pulses to each other in this giant connection of the brain. Okay, so let's first talk about structure. So as I said previously, the neurons in the brain are all connected to each other in very specific patterns. And it's those specific patterns that make up the brain, and the function of the brain depends on all of those patterns. There's a word for it. It's called a connectome. So the connectome is a complete mapping or wiring diagram of every single neuron in the brain. And it's the goal of connectomic analysis to try to map out that wiring diagram, to understand exactly which neurons are connected to which other neurons in the brain. And the question is, can we figure out that mapping? Can we uncover the connectome? And the answer is, it's already been done for one animal, a tiny worm called C. elegans. In a major scientific breakthrough in 1986, J.G. White mapped out all 302 neurons of this tiny worm. All of the connections are now known. This is a computer model that's showing you the 302 neurons in the worm and the connections between them. But we want to move beyond the worm. We want to go and try to see if we can figure out what the connections are in higher level organisms, such as maybe a rat that has millions of neurons, or even a human that has about 86 billion neurons in its brain. But how can we do this? Well, I'm going to show you a video clip in a second of a group that has found the connections of a very small piece of rat brain. And to explain how they did this, I want to use an analogy of this landscape. So you can imagine the picture on the left is showing this landscape with mountains and hills and the lake. And if we want to build a computer model that describes the height at any point on this landscape, you could imagine hypothetically slicing it parallel to the ground, picking up each slice and stacking them on top of each other to build the representation on the right. Well, that's what these researchers did. They took a small piece of a rat brain and they sliced it into very thin slices and then imaged each slice using electron microscopy. And then they stacked all those images together and traced where all the neurons are passing through this stack of images. So let's go to YouTube and watch this video. So here we're zooming in into this electron micrograph.
Here we're moving through the stack of brain slices, like a flipbook. The team took these still images and crunched the data to rebuild the piece of brain tissue. It's from a bit of the mouse brain that receives information from the skin. Here are all the neurons which feature in some way in the speck of brain the team focused on. But they were really only able to analyse this much. They focused on 2 brain cell branches that interact in this portion. You can start to make out the components here. In red and green, the 2 main cell branches. Then, all the other neurons that travel through the area at some point. Here are all the synapses, the connections that neurons make with each other. Here are the inhibitory connections, which dampen down activity. And not forgetting the non-neuronal cells, like these glia, which support the neurons and help them send messages to each other. All in an area smaller than a dust mite.
All of that in an area smaller than a dust mite. So I want you to appreciate the complexity of this tiny little piece of brain and all of the neurons and components of the brain that are coming together at this one little point. Now, what if we wanted to do something like this for a human brain? Well, to lower your expectations, this has not been done. But I want to go through the process of seeing what that would look like, just to show you how large the brain really is. So let's start with dimensions. So the brain is approximately a cube, about 10 centimeters on a side. That's not exactly true because your brain is longer as you go from front to back, but this will be fine for an approximation. So what we would have to do is we would have to slice the brain into various thin slices and take electron micrographs of each slice. Each of those slices is an image, and it would be about 10 by 10 centimeters. Now that's also not exactly true because as you go towards the edge of the brain, the images will get smaller and smaller. But this approximation is fine. So this image on the right is the same as the cat image in the respect that it's made up of pixels. And we have to convert it into zeros and ones so that our computer can understand it. But how many pixels are in this image of a single slice of our brain? Well, in order to figure that out, we have to know how big each pixel is. And it turns out that if your pixel is 30 nanometers, a nanometer is a billionth of a meter. If it's 30 nanometers long, that's of sufficient resolution for you to be able to image all of the cells and all the connections between the neurons. Now, this is enough information for us to figure out how many pixels are in a single slice micrograph. What we have to do is we have to take, we'll do this first for a single row of pixels along the image. We have to take the length of that row, which is 10 centimeters, and divide it by 30 nanometers, and then convert the units. So we start with 10 centimeters, convert units to meters, divide by 30 nanometers. There are a billion nanometers in a meter, and that gives us about 3 million pixels along one row. That's about as many pixels as in the cat image, or any typical image that you might take with your camera, your cell phone camera. Now, in the entire image, we have to square that. So it's about 3 million squared pixels in the entire image. But we have many images. We have many slices of the brain. Each slice being 30 nanometers thick. So now, instead of pixels, we're going to talk about voxels. And a voxel is a volumetric pixel. So it's a single unit in a volume that we have to divide our brain into. So how many voxels are in our brain? Well, in 2 dimensions, we said it was about 3 million squared. For the entire volume, it's going to be about 3 million cubed, which is a 36 with 18 zeros at the end. It's a huge number. And if we were to assign a single byte to each of those voxels to record it in a computer, that would take up 36 exabytes of space. What is an exabyte? Well, let's think about units of measurement. And these apply to anything you want to measure, length, mass. But we'll be talking about bytes, the storage in the computer. So kilo is the first one, it's 1,000. And as we go down each line is 1,000 times bigger than the line above it, adding 3 zeros at the end and adding 3 to the exponent on the left. So a megabyte is about the amount of storage it takes to store a cat picture. As we go down, a gigabyte is about the amount of storage it takes to store a single movie, an hour-and-a-half long movie. A terabyte is about the amount of storage you might have in your laptop. Now, if you're working on a supercomputer, like one of the ones that Lawrence Livermore, you might hope to operate on petabytes of data. We're moving towards the exascale, but we're not there yet. Our brain image, to image a single brain, would be in the exabyte scale. So even if we could do this exercise, we wouldn't be able to have anywhere to even put all this data. Right? It gives you a sense for how large of the structure of the brain is. Okay, let's go on and talk about function. So recall, function is the activity of the brain. We just saw how complex the structure is, the interconnections between all the neurons, but those neurons are talking to each other and transmitting signals to each other. So when does a neuron decide to send a signal down its axon? Well, let's talk about this behavior. The behavior of the activity of a neuron is an all or nothing behavior. Either there is a signal on its axon or there's nothing. It's sort of binary. And we'll call this spiking behavior. So a neuron will decide to send a spike down its axon when there's a large enough stimulus coming into the neuron through its dendrites. When all of the signals coming into this one neuron is large enough, reaches a certain threshold, then this neuron will send a spike down its own axon, and through the axon terminals, it then connects to the other neurons. The signal then is transmitted to the other neurons that it's connected to. Now, while we understand very well how a single neuron works, the truth is that when you put it in such a large, complex structure, 86 billion neurons in the brain, we don't understand how that large structure works. A group in Europe, I'll show you a video now, designed a computer simulation to try to understand what the function looks like, what's the behavior of the function when you have a large population of neurons. What they did is they took a large collection of neurons, and they programmed them in a computer with all the rules that we know about how a single neuron works. And they made all the connections inside the computer. Then they applied a stimulus to it and saw where those signals were propagating through the neurons. So let's go to YouTube and watch this clip. So here you're seeing each streak of light is a single axon of a neuron. So I want you to notice in this column of neurons, this sort of rhythmic type of behavior, where you see this light kind of pulsating through this collection of neurons. We'll come back to this later. But now I want to think about what it would take to store all of the function, or all of these spikes that are inside of your brain in a computer so that we can analyze them. Well, first, let's start with some numbers. You have about 86 billion neurons in your brain. And we're going to assume that each one is spiking once every 10 seconds. Now, some of them will be activated much more frequently and some of them less, but let's just assume on average once every 10 seconds. Now, before we can go further, we have to go back to bits. And I want to ask a question. And that question is, how many combinations of 2 bits are there? Now, this seems like a very abstract question, but this is related to the question that we're actually interested in. So if you have 2 bits, how many combinations are there? The answer is 4. 2 to the second power, 2 times 2. And here they all are, 0, 0, 0, 1, 1, 0, and 1, 1. So 2 bits can represent 4 different possibilities. You get 4 labels, 4 unique labels that you can use to label 4 different things. Okay, what about 3 bits? How many combinations of 3 bits are there? The answer is 8. So 2 to the third power, 2 times 2 times 2. And here they all are from 0, 0, 0 to 1, 1, 1. So with 3 bits, you get 8 labels. You get 8 different combinations. You can label 8 different things with 3 bits. So now the question that's related to our problem, how many bits can represent 86 billion possibilities? Because we have 86 billion neurons in the brain. This question is a little bit hard, so let's think about this. If you had 36 bits, 2 to the 36 is about 64 billion. A 64 billion is not enough. We would have about 20 billion neurons left over without a unique label. With 37 bits, you double the 64 billion to get 128 billion. That's more than 86 billion, but that's fine. We'll have some labels left over, but at least each of the 86 billion neurons in our brain gets a unique label. So every time a neuron activates, we're going to specify which neuron activated with a string of 37 bits. Okay? So back to our problem. We have 37 bits to tell us which neuron it was that spiked, and we're going to use 32 bits, or 4 bytes, which is floating point precision, to tell us what time it spiked. So every time a neuron spikes in your brain, we're going to need 69 bits to tell us which one it was and at what time it spiked. So now we can figure out the data stream, or how much data your brain is generating. So for every second, we have 86 billion neurons. It takes 69 bits, as we just said, once every 10 seconds on average. And there are 8 bits in a byte. So under this approximation, it's 73 billion bytes per second, or 73 gigabytes per second. Now, how big is that? Well, a movie streams at about 800 kilobytes per second. So your brain is streaming the equivalent of 90,000 movies simultaneously when you're at all times. Right now, you're probably doing a little more because you're so engaged with this presentation. So it's an incredible amount of information that your brain is generating. But we can't record all of the neurons. It's just too much. But we can record some of them. We can use devices to record some of the electrical activity of the brain. So through a technique called electrophysiology, which is the general study of recording electrical activity from the body, we can record the activity of the brain. So on the left, I'm showing a technique called EEG, or electroencephalography. And this is a set of sensors that you wear kind of like a hat. And what happens is, when the neurons inside your brain are working, they're emanating this electric field that actually propagates through the skull and these sensors can pick up very weak signals representing the activity of your brain. And this would take about 4 gigabytes per day, is the amount of data you can get from one of these sensors. Now, through our collaboration with the University of California, San Francisco, Edward Chang and his group, we are recording data from called electrocorticography, which are implanted devices. This is a real photograph. These devices sit on the surface of the brain, and they record very detailed information pertaining to the electrical activity of the brain. This is an amazing opportunity that we have to get a unique glimpse into how brains are functioning from living human patients. So how does this work? So the patients have intractable epilepsy, and they're awaiting surgery. So they're in a monitoring ward for about 7 to 10 days at a time, and the sensors are implanted at the beginning of their stay. So through the sensors, we're able to measure the function of the brain. Now, we focused our study on emotional affect and mood. So we want to understand, what does the brain look like? What's the activity of the brain when the patients are in different emotional states? So think about this when you're watching the movie, because this is a very direct connection to the themes of the movie. Well, we're measuring the emotional state of the patients through a video camera that's positioned in the monitoring ward. So the video camera is picking up individual cues of emotional affect. Things like laughing, crying, smiling, saying something positive, saying you're in pain, saying something negative if you're unhappy perhaps. And through those individual time points, individual moments of emotional affect that they display throughout the course of their week-long stay, to try to understand what their brain is doing. So let's step back for a moment and talk about emotion. So as I've said, emotions are individual displays. Like they're short durations. And they're intimately connected with how the brain works. Humans have had emotions for a very long time. And they're there for survival reasons. Okay, so for example, when you're afraid, that might help you escape from danger. It's your body giving you a cue, right, that it's time to run away, for example. When you're sad, that helps with attachment to your family. When you're happy, that gives you an opportunity to rest. Okay? So these are all cues that have been developed over many, many, many years that help us perhaps increase our likelihood of having a successful life. Now, a mood is a long-term state. So just like weather and climate, right? Weather is a short-term duration. It could be raining today and sunny tomorrow. But climate is the change in weather over decades of time, right? And those are much more gradual and smaller. Just the same thing. You could be happy today and sad tomorrow maybe, but your overall mood is the composition of many, many instances of emotional affect over long periods of time. So what can happen, for example, if you're consistently afraid, maybe developing a phobia, right? So we start to talk about mood disorders. If you're consistently sad, you could be developing depression. Now, what could possibly be wrong with being happy all the time? Well, perhaps that's the basis for addiction. Right? If you're doing something that consistently makes you happy, like playing video games, just kidding, not really, then it makes you want to continue that activity and it could potentially form an addiction. Okay, so let's go back to our problem. We're recording data from these ECOG arrays that are implanted in our patients. We have about 100 sensors on each patient, and each sensor is recording a signal. Okay, so you're seeing two different signals depicted on this slide here. And what we would like to do is to understand what these signals look like when our patient is in either, say, a happy state or a sad state. And we're able to measure a signal when we have large collections of neurons that are activating simultaneously or coherently. If there's an oddball neuron that's spiking here and there, the signal is too weak for us to measure it. Okay? So I want you to think back to the video with the computer simulation. And you saw this large column, this large population of neurons that act where you had this propagation of this pulse, this wave-like activity. Large number of neurons that are activating simultaneously. When that happens, we can measure it. And that's a wave-like activity. So we want to make sense of the waves that are contained within the signals that we measure. Waves can sometimes be slow, or they can sometimes be fast, and everything in between. When we talk about signals, we talk about low frequencies when the distance between the peaks is large, or high frequencies when the distance between those peaks is small. And we have everything in between. So what we'd like to do is compute the composition of these frequencies in the signals that we measure, which then tells us something about how the neurons are activating in the brain of the patient. So using a technique called the Fourier decomposition, this was discovered by Joseph Fourier in the early 19th century. It's actually a technique that underlies many of the technologies that we use today, communications, audio analysis, etc. We're using this technique to take the signals that we record and break them down into the frequencies. Okay? So by doing this, we can understand which frequencies are contained in each of the signals. So this bar graph is showing you a depiction of that, and maybe there's less of a lower frequency and more of a higher frequency. We have the entire sort of distribution of all of those frequencies. And now what we'd like to do is look and try to see if we could figure out which frequencies are contained in our signal when our patient is in a happy state or a sad state. So we'll talk about emotional frequencies, which is the joke, because the frequencies aren't really emotional. But we're going to talk about which frequencies are occurring under which emotional state of the patient. So what we'll do is we'll use the video camera to extract those times when a patient is exhibiting a positive emotional affect, being happy, and then we'll take the signals that have occurred during those happy times, and we'll compute the frequencies. Okay, so we now have a model that tells us what the frequencies are when the patient is happy. Now those frequencies also depend on where in the brain we're sensing, because remember we have 100 different sensors in the brain. Then we'll do the same thing for the signals recorded when our patients are in a sad state, a sad mood. And that gives us a model or a description of what the frequency contents are when the patient is in a sad emotional state. We can then use those two models to create a scale. We can quantify how happy or sad the patient is on the basis of their brain signals alone. So the way we do that is we can extract a new signal, and we don't know if this particular signal is coming from a happy or a sad state. And we can compare it to our two models that were developed using other data from the same patient. After we compare this new signal to the two models, we can generate a scale in order to quantify how happy or sad this patient is. We can then validate the model by determining how well our model predicts whether the patient is happy or sad. And we can see how well our model does as a function of location in the brain. Okay, so this figure is showing you a 3D model of an actual patient's brain, and each dot here is a location of the sensor. And they're color-coded from blue, which means a poor detection of emotional state, to yellow, which is a good detection of emotional state. So you can see here which locations we're doing a better job of detecting their emotional state. So we can use this to better understand and perhaps keep track of the emotional state of the patients, but more broadly, we can use the incredible access we have to this precious data to try to better understand how emotions occur in the brain and what the effect of them is. So this 3D model is showing you all of the sensors for this particular patient, and this can also be used to plan surgery, right? So, for example, a surgeon doesn't necessarily want to go in and affect a certain region if it has a high likelihood of disrupting your ability to process emotions. So to wrap up, the brain is immensely complex. I hope you got a sense for that in this presentation. And it's so complex that we need computers, in some cases very large computers, to understand how the brain works and decode its functions. Through this research, we're gaining new insights into how emotions are represented in the brain and how mood comes about over longer periods of time. And ultimately, our motivation and goal is to improve the outcomes of patients that are affected by a brain disorder or otherwise in a hospital setting. Thanks for your time. I hope you enjoyed the presentation, and I hope you'll enjoy the movie.